Hello, everybody. People are still entering the attendees list, so I will leave it for a couple of minutes. We still are 2 minutes to 2 o'clock where I am. Okay, we have come up to the full hour, and I would like to start the last session of the annual UFAW conference. My name is Birte Nielsen. I'm the Assistant Scientific Director at UFAW, and I will be chairing this session. A few practical things for those who haven't joined us before. You will ask the questions in the question box, and you will find in the chat some information about certificates of attendance and information about a voucher to get a discount for 20% of books at the UFAW Wiley Animal Welfare Series. And also there will be a link, or there is a link already to the recordings, not only of yesterday, but also all the recordings from this morning. And this session will also be posted there later on today. So thank you very much for joining us for this last session. It's been 2 really action-packed days, and I will start by telling you the winner of our poster competition. As you know, you could all go and look at our posters and leave comments and like the ones you thought were the best ones. And as of this lunchtime, we have done the tally. And I am very pleased to announce that the winner of our poster competition is poster number 89 by Christina Ramos and colleagues from the University of Saskatchewan, Survey of Canadian Producers on Factors Related to Sow Mortality and Culling. So please go and see poster 89, and congratulations to Christina. The prize is very kindly donated vouchers from Wiley that you can use to buy, hopefully, some of the UFAW Animal Welfare Series. We will contact you after this event with the details. We also have 2 runner-up posters, and these will be shown in the next slide. That is poster number 33 and 46 by, respectively, Samuel Durazuro and Ziun Yasera. And you can, as you know, still watch all the posters on the website using the handouts, the link in one of the handouts that you can find in the box connected to this session. And with that, we go to the first speaker of today's session, and I'm very, very pleased to say that it is one of our early career researchers of the year, Dr. Jamie Ahloy Dallaire from Quebec in Canada, and he will be giving a talk on identifying and raising adaptable animals. Here you go.
It's truly a pleasure, an honor, and a privilege to speak to you today and to accept the Early Career Research Award. I want to start by thanking UFAW for this amazing opportunity, and of course by thanking my mentors, Georgia Mason and Joe Garner, who nominated me for the award, and more importantly, who gave me unwavering support from day one. And of course, I also want to congratulate my co-recipient, Dr. Chou. I'm going to start with a very brief outline of my research so far, without going into great detail about the results, and then tell you about the research program that we're currently putting in place at Université Laval. My master's research was about differences between individuals, in terms of how they react to being given environmental enrichment. We were interested in why some animals can spend their entire lives in inadequate conditions and then be rehabilitated successfully, while efforts fail for other individuals. I actually received a UFAW Animal Welfare Student Scholarship 11 years ago to run a short project on BioBear rehabilitation, where I was looking specifically at how physically disabled individuals were adapting to their new enriched environments. But the bulk of my project was run on a different carnivore, the American mink, which is a more convenient model species. We worked with older animals who had spent their whole lives in standard farm conditions and assessed behavior and welfare before and after transferring them to enriched environments. Our primary outcome measure was stereotypic behavior, which is a type of abnormal behavior associated with poor welfare. And we wanted to know why our mock rehabilitation would eliminate stereotypies in some individuals and not in others. Surprisingly, we measured lots of things, but none of them actually predicted which individuals would best recover. Whether that be how predictable their stereotypies were in form, how each animal performed on operant tests that assessed impulsivity and cognitive flexibility, how much time each individual spent manipulating enrichment objects, or how motivated they were to access these objects. But we did discover that the most stereotypic animals showed considerable recovery, even though they barely ever used the provided objects. Enrichment use was much more frequent in animals who were not that stereotypic to begin with. For my PhD, I worked on a different type of behavior that's also often defined as apparently useless: play in juvenile mink. Play seems to have no obvious goal. For example, there's no clear way in which doing this helps animals feed themselves, and it probably seems useless because its benefits are realized in the long term. For example, in our longitudinal research, we showed that in males, the juveniles who spent the most time in play fighting go on to show increased sexual competence as adults. So the most playful males later copulate for the longest durations, which probably gives them an advantage in sperm competition. And that's consistent with the hypothesis that playing helps young animals develop certain skills that will prepare them for future challenges. But what I also investigated, and was especially interested in, is the effect that play might have on the emerging personality of young animals, particularly when it comes to how they react to dangerous, stressful situations. And I'll talk about that in more detail in a few minutes because it's an integral part of my ongoing research program. Finally, during my postdoc, I worked primarily with lab mice, and my main project involved designing and building an automated system that would let us assess pain in a valid, efficient, non-invasive way. The system consists of a series of drinkers that can be integrated into any mouse cage to control and monitor voluntary intake of analgesics by individual mice in group living settings. The idea is that if mice are in pain, they will learn to consume effective analgesics, and that they'll consume larger doses if the pain is more severe. And that's as much as I have time to say for now. Let's turn to the research program that we're starting up at Laval. We're working with piglets, which I initially chose as an excellent model species to carry forward my research on play. But you'll see that there are some clear links between this research program and what came before it, not least because all the projects are centered around the same concept of robustness, which was also the case in my mink research, even though I wasn't really thinking about it in those terms at the time. Robustness is the capacity to face and overcome challenges without experiencing major negative impacts. For example, an animal who's robust with respect to physical health would not be severely affected by exposure to pathogens. The animal could be robust by being resistant, which would mean they don't get very sick, and or by being resilient, which would mean they do get sick but they recover rapidly and completely. Our research program aims to understand the potential determinants of robustness with respect to animal welfare in piglets. And I'll tell you about 3 projects focused on different types of determinants, and I'm summarizing them at a very high level, so feel free to ask me about the details afterwards. Starting with internal factors at the level of personality and genetics. This project is inspired by things we know about human welfare. There's a large literature on what psychologists call subjective well-being, which they assess by asking people questions like, on a scale of 1 to 10, how happy do you feel? Or how would you rate your quality of life? And as someone who works with nonverbal animals, I am not jealous of this. The literature on subjective well-being teaches us very interesting things. First, that individual differences in well-being are stable over time. So if someone tells you that they feel 8 out of 10 today, they'll tend to give you a similar score if you ask them again in a few years. Second, external circumstances like income level, family status, don't affect subjective well-being as much as we might think. Even major life events have mainly short-term effects. When someone loses their spouse, loses their job, becomes paraplegic, or when they win the lottery, their subjective well-being can be affected dramatically, but after a year or two they tend to adapt to their situation and revert to their initial baseline level of well-being. In other words, this means that in humans, subjective well-being is robust. It's robust because it's determined not directly by external circumstances, but actually by the manner in which each individual perceives and interprets those circumstances, and that's an aspect of their personality. Finally, the literature also shows us that there's a genetic component to subjective well-being, and that leads to the question, does animal welfare work this way? And would it be possible to artificially select for robustness with respect to welfare? Now I hear you saying we cannot measure subjective well-being in animals because they don't talk, which is true. So in this project, we're instead going to use several different behavioral and physiological welfare indicators. And among these, the one that to my mind gets closest to subjective well-being is judgment bias. Very briefly, this is assessed by first teaching an animal to associate one stimulus with a reward, like food, and another stimulus with punishment, or no reward. And once the animal has learned to react differently to each of these stimuli, we start prompting with intermediate stimuli to see whether the animal interprets this ambiguous situation optimistically or pessimistically. Does the animal react as if the stimulus is most similar to the positive one or to the negative one? And this test is well validated in many species, including pigs, as a good way of assessing overall mood. The first objective of the project is to develop an automated apparatus to train and test nursery piglets directly in their home pens. Just like in my postdoctoral project on mice, this would allow us to collect data efficiently, continuously, non-invasively. And as you can see from the cartoon schematic from a recent project meeting, we are still in the early design stages. Our first animal trials will aim to refine the apparatus and the protocol until we know that we have something the animals can successfully be trained on and tested on, and that the animals will continue to show interest and good performance over several weeks of repeated testing. The second objective will be to test whether the automated task is a valid measure of judgment bias and overall mood. To do that, we'll compare results between animals exposed to treatments that already have documented effects on mood and on non-automated judgment bias measures. And finally, our last objective will be to test if individual differences in welfare, assessed by judgment bias and by other behavioral and physiological indicators, are stable over time, and if they're at least somewhat insensitive to external circumstances, as in humans. Halfway through the nursery period, some piglet groups will be switched between treatments. So for example, going from standard to enriched, so that we can test whether the happiest piglet in each pen at the start of the nursery period is still happiest at the end, and whether that still holds even if the group has been moved to new living conditions. At the end of the project, we should have a good idea of whether these individual differences are stable over time and robust, and if so, we'll also have an automated tool that would allow us to efficiently assess this phenotype on a larger scale, which will be necessary for subsequent projects on the genetic aspects underpinning these differences. Our second potential determinant is a developmental one, the expression of play early in life. The main hypothesis that we want to test is that play is a way for animals to learn how to cope with surprising, unexpected, dangerous situations. According to this hypothesis, first put forward by Marek Spinka and company, play gives animals a generalized form of preparation for a wide variety of unexpected situations. You can see where this idea might come from by looking at these pictures and thinking about how silly these animals look. They're jumping off of logs, they're twisting their bodies and heads in midair so they can't actually see where they're going to land. They're trying to pin down their partners by climbing onto them on all fours, which is ridiculous because as soon as the bottom rat moves, the top rat will go flying. And the idea is that when the animals play, they seem to be deliberately putting themselves into precarious situations where they'll lose their balance, fall down, be surprised by an opponent, because the function of play might be to learn how to react in these types of situations. And if that's correct, then playing would help animals cope more effectively, both physically and emotionally, with this type of situation in the future. And if that hypothesis is correct, that would mean that playing equips them to cope with the types of stressful events that can be facts of life for farm animals. And if so, the benefits of promoting play in juveniles would be 2 for 1, in the sense that this would lead both to positive emotional experiences in young animals and to buffering against later negative experiences. One of the things we want to do around this hypothesis is longitudinal studies, similar to what I've already done in mink. We'll be testing piglets' reactions to various different stressors, including contact with human handlers, truck transport, and introduction to a new social group. We'll run the tests both at the beginning and at the end of the nursery period, while observing how much each individual engages in different types of play in the interval. And that will allow us to test whether animals who play more correspondingly also show larger improvements in terms of their reactivity to stressful challenges. We'll also use a second complementary approach, where we will first test whether various different modifications to housing conditions can reliably stimulate or inhibit play for the whole duration of the nursery period, which would mean that some animals have been experimentally made to play more or less than others. We'll then be able to use the same outcome measures as in the previous objective to test whether experimentally inducing play can have long-term benefits in terms of buffering against stressful events. And we'll also note a little bit more about how we might implement this in practical farm settings. The last potential determinant is social dynamics within piglet groups. We're specifically interested in knowing how resistance to infectious disease can be affected by social support. Social support is the phenomenon where having positive social interactions and relations helps buffer stress reactions. In humans and lab rodents, it's well documented that through its impacts on stress physiology, social support also has downstream effects on physical health. For example, humans and rodents who maintain relationships with conspecifics have been shown to have lower risks of mortality from cancer, slower tumor growth, faster wound healing, and even better resistance to infectious disease. But the impacts of social support on physical health haven't been studied as extensively in farm animals. Our project on this question is going to take place in what's called the natural disease model. The CDPQ, the Quebec Pork Development Center, has been running this model at their research nursery since 2015. And briefly, what it is that one section of the nursery is deliberately maintained in poor sanitary conditions, mimicking a commercial environment with high disease pressure. So the piglets are exposed to a cocktail of different viral and bacterial pathogens. And previous research has shown that piglets with physical and social enrichment show increased resilience to porcine reproductive and respiratory syndrome virus, which is the main pathogen that affects these piglets in the nursery. But we can't necessarily tell from this research if that beneficial effect was due specifically to the social environment of the piglets. Almost 2 years ago, I went on a tour of the CDPQ nursery, and they told me about the natural disease model and how they'd established it to identify genetic factors associated with resistance to infectious disease. And they also mentioned almost offhand that they'd been filming all the piglets continuously. So they have this vast trove of archived video of group-housed piglets exposed to infectious challenge, and I immediately decided to worm my way into this project, to get my hands on that video and test the hypothesis that if some piglets don't get sick, or if they get less sick than others, it is in part because of differences between social groups in terms of the relative importance of positive and negative social interactions, and because of the position of each individual within their group. The first objective of this project is to characterize that relationship between social dynamics and resistance to infectious disease. And we'll use the video data from historical cohorts and data from new cohorts, which are part of a collaborator's project. The piglets in the new cohorts will also be equipped with lightweight sensors attached to their ear tags, which will give us continuous location and accelerometry readings. With my colleague Eric Paquette, who's an expert in big data and machine learning, we'll work together to automate analysis of certain social behaviors using just the sensor data, and also to make video observations more efficient by using the sensor data to target moments when we know the animals are interacting in some fashion. And from these measures of social behavior, we will construct social networks of both positive and negative social interactions. And then we'll test whether network parameters correlate with health status and performance outcomes. And so we'll see if more harmonious groups or individuals with close or numerous friendships are more resistant to the infectious challenge. As a second objective, for which we'll use new cohorts with smaller groups of piglets, we will also test whether interventions that improve social interactions also provide health benefits in this model. The treatments that we chose all have documented benefits, at least when it comes to reducing aggression, but we know less about their potential effects on positive social interactions. If we do find that steps taken to improve social dynamics also make piglets more disease resistant, this will be another case of 2-for-1 benefits, because the same interventions will impact both the mental and physical components of well-being. I want to end with a quick word about why I'm so interested in robustness. I think robustness with respect to animal welfare is something we must come to understand, that we must promote, in large part because farming practices are constantly evolving. The ways in which animals are housed and raised today bears many differences with how this was done 40 years ago, and that was different from what came 40 years earlier. Every way of raising animals imposes on them a set of different challenges, which aren't the same from one system to another. And that means that the animals who are best adapted to one particular way of life, based on their genetics, their development, their personality, their social group, well, these animals aren't necessarily those who are also best adapted to other ways of life. 40 years from now, things will have changed once again, and right now we're not in a position, I think, to confidently predict what animal farming will look like, which means we don't yet know what the animals should look like so that they can maintain optimal welfare in those conditions. But robustness is a generalized capacity to face all sorts of different challenges. And so by definition, it's a quality that's not specific to one particular way of life. And that's why I think that promoting robustness in farm animals is an important way of future-proofing our practices, of getting a head start by developing, right now, animals who can maintain better welfare no matter the conditions they find themselves in. To conclude, I'd like to thank my research team, a group of students I've displayed from left to right according to the potential determinant that they study, and Annick Boileau, my first graduate student, whose project has nothing to do with robustness, but who makes valuable intellectual contributions to that research program. Thanks also to our collaborators, to our research partner organizations, and of course our funders. Thank you for listening, and I'll be happy to take any questions that you may have.
Thank you, Jamie, for this very, very interesting talk. And there you are. We are running a little bit over time, but I took some of your time in the beginning for announcing the winners of our poster competition. So I will allow this to run a little bit over the scheduled timing. Lots of questions coming up for you. Rachel Casey wants to know that you mentioned augmentation or automating repeated judgment bias measurements. How are you planning to overcome potential issues of learning about ambiguous stimuli, which might happen over time?
Excellent question. That's one of the things that we've struggled with, that Gwendolyn Garoui, the student who's working on it, has been reading a lot on. And there are many potential things that we could do that might help, and so we'll be trying them and then seeing what seems to work. So for example, one of the things that we might do is to make the outcome of a response to an ambiguous situation actually random, so that if the piglets are learning about what happens when they react to the ambiguous situation, they're actually learning that sometimes it produces the positive outcome, sometimes it produces the negative one. And of course, related to this, there's the problem of keeping them engaged long term. So that's the other big big challenge we have to face. But yes, I think teaching them that the outcome is random is probably our first and best bet.
Thank you. Sorry, I'm juggling my mic here. Another question is it known in humans how early in life, this is from Lisa, how early in life this stability and assessment of well-being occurs, this personality that you're talking about? Is it from childhood already, or is it only in adulthood that it stabilizes? And would you expect that young pigs would already show this kind of stability in mood at the time that you are measuring it?
That's also a good question. So most of the studies on subjective well-being that I've read in humans are done in adults. A lot of them are twin studies where twins are separated at birth, raised in different environments. This is the parts on genetics, and then they're found to have similar levels of well-being, even if their external circumstances are quite different, but they're assessed as adults. And actually, this question mirrors one of the questions that we got from evaluators when we submitted the grant, which was, shouldn't you work with older pigs? Because that's when well-being might be more stable. They're going through a lot of changes when they're young. And I kind of see this as maybe a benefit of working with juveniles, in that if we see that well-being is robust over time at that age, even at that age, then that's a good indicator that it might be robust also at other ages, because I think we're kind of starting in the most challenging circumstances. But certainly, yes, that is a possibility that they're going through childhood and the start of adolescence at that point, and things change a lot.
Thank you. Another question, a mixture of Stephen is asking, how do you make animals play more? And I have an additional, I suppose, thing to that question is, you talk about that pigs or animals that play more are more robust, but is this really a sort of is it a correlation, or can you play too much? Can you be a risk taker that is then somehow exposing yourself to more risks than necessary, or is it simply the more you play, the more robust you are?
Okay, I'll start with the second question first. I don't think we know. I mean, certainly there are some documented effects, especially in wild animals, of animals who play too much and actually pay the cost of the risk. For example, I believe it's harbor seals who are going out and riding the surf and doing more locomotor play for larger periods of their day are actually at more risk of being predated upon by, I think it was sharks in that study. So there are real costs potentially to playing too much. But in terms of, let's say, a longer term developmental cost, does an animal who plays too much as a young juvenile end up with some sort of negative effects long term? I've never seen anything about that. It could be. And then in terms of how do we make animals play more, so that's something that we need to test and try. I spent a long period of time during my PhD trying to do this in mink, and it was very challenging. I think going to pigs, one of the reasons that I consider them a better model for this is that we already know more about how to make them play. For example, we know that providing them with novel enrichments, with a rotation, with more space is probably one of the effective ways of doing it. We're pretty confident that dividing up their environment so that they have the same overall space, but their movements are restricted. For example, if they can't go for a long run, would be a way of inhibiting play if we wanted to go that way. And the goal really would be to come up with several different successful experimental manipulations that will make them play more or less, so that we can see if they all have the same effects on the development of temperament. Because if so, then we can be more confident that it's not an effect that's specific to the type of enrichment that we gave, for example, but it's actually something common to all manipulations that affect play. But yeah, confirming that is kind of our first step.
Yeah, yeah. There are lots more questions, but we are running out of time. I was very pleased to hear, Jamie, that you had received the UFAW scholarship 11 years ago. So UFAW is probably unashamedly going to take, the responsibility for your career and all the success that you have. Thank you very much, Jamie. And we will now go to our next speaker, which is Carly O'Malley, who will talk about driving organizational change in welfare practices, a stakeholder focus group approach. Thank you.
Within research environments, the 3 R's is the guiding ethical framework for addressing and improving animal welfare. The 3 R's include replacement of animal models with alternative methods, reduction of the number of animals used in research through proper experimental design, and refinement of animal care and research methods to minimize pain and distress and optimize welfare. For this talk, I will be focusing on the 3rd R, refinement. Refinements can include using low-stress handling techniques when interacting with animals, using positive reinforcement training methods to encourage animals to cooperate with husbandry and study procedures, providing adequate housing that encourages natural behaviors, and overall ensuring animal comfort throughout their time at the animal facility. Refinements in the research environment are key to maintaining and improving good animal welfare and for addressing ethical concerns of working with animals in science. Refinements are not only important for animal welfare, but also for the quality of the research data. Research organizations need to continually evaluate their animal care and use practices to ensure modern evidence-based practices are being used to minimize pain and distress and provide animals positive experiences. Within Charles River, we work with many species of animals across 99 active sites in 23 countries. There are multiple business units, each with their own objectives and goals. Implementing refinements in a contract research organization such as this can be difficult. It is a large, complex business involved in a wide variety of projects with varying research practices and requirements based on the study sponsor. The facilities are located around the world, meaning there are a wide variety of regulatory and compliance requirements. There are also differences in cultures surrounding animal care and use across countries. When it comes to implementing refinements, there can sometimes be a reluctance to do so without a legislative or regulatory mandate. A lack of flexibility when making changes at a corporate level that impact all sites may overwhelm business leaders and site heads and study directors and those responsible for establishing research contracts. Or it may lead to non-compliance if the mandate is too strict or if personnel do not see the benefit of the refinement. There also may be a lack of resources, human, financial, or otherwise, to immediately implement refinements or differences in priorities across sites and countries. Promoting evidence-based refinements and encouraging communication and collaboration between facilities may lead to greater willingness to implement changes and more site-level innovations for improving welfare. One way to achieve this is through stakeholder focus groups. Stakeholder focus groups are commonly used in animal science industries to better understand attitudes towards different policy changes or welfare issues, identify challenges within the various industries, and opportunities for promoting welfare-driven changes. Stakeholders often have conflicting opinions on what the welfare issues are and how to improve welfare. Focus groups provide opportunities for stakeholders to discuss differences of opinion, collaborate, share knowledge, and arrive at decisions on how to move forward in an inclusive way that has buy-in from all the stakeholders involved. Stakeholder focus groups should be built on the following principles: reflexivity, responsiveness, revitalization, resilience, and relational capital. The principle of reflexivity is to take multiple perspectives into account. Responsiveness refers to the ability to respond to changing demands from stakeholders. Revitalization is the ability to respond to conflict by redirecting stakeholders to the common goal. Resilience refers to maintaining flexibility to unpredictable challenges, and relational capital is the principle of establishing and maintaining collaborative relationships between stakeholders. By using these principles, the goal is to build a collaborative network of stakeholders that can continue to work together to improve welfare as we learn more about what animals need and as research priorities and practices continue to evolve. For the past 50 years or so, pigs have been increasingly used in research. Pigs are an important food source worldwide, so pigs are commonly used in agricultural research. But pigs also share a number of similarities with humans that have made them an important biomedical research model. Pigs are similar to humans in their anatomy, physiology, diseases, and metabolism, just to name a few model areas. Pigs are particularly useful as surgical models and in dermatological and infectious disease studies. Mini pigs were developed as a more convenient and economical model to house in a research environment, but conventional large breed pigs are still often used. Pigs can be a difficult species to work with due to their large size and their strength. Compared to other research models such as primates and dogs, their welfare needs are less well described. In North America, pig housing and husbandry practices, especially those for mini pigs, are often adapted from dogs rather than providing housing and handling techniques specific to the biology of pigs. For example, small pigs sometimes may be housed in modified dog cages. This may lead to injury if pigs try to escape from the enclosure, as pigs are not as athletic or agile as dogs. The flooring and other pen elements are important considerations, since suboptimal surfaces may lead to issues for pigs navigating their environment. Like other species, pigs are often restrained manually for study procedures, which can be stressful and dangerous for study personnel and animals if the animals struggle or are inadvertently dropped. Some practices used for research pigs can increase the risk for injury and even result in the need for animal euthanasia. When thinking about optimizing the care and handling of pigs in a research setting, it's important to remember that pigs are highly intelligent animals that will respond well to training, but training habituation and desensitization are still not commonly used with pigs. General guidelines are available for the care, husbandry, and space requirements for pigs, but these are often minimal standards, and otherwise there are no internationally recognized standards for housing and caring of research pigs. To address the need for more formalized recommendations and capitalizing on the lunar year of the Pig Welfare Working Group was formed. The goal of the Pig Welfare Working Group was to establish global guidelines and best practices for research pigs within Charles River, and the aim was to recruit a diverse group of stakeholders to provide input into the recommended refinements. The Pig Welfare Working Group is formed and organized by Global Animal Welfare and Training, which has corporate oversight of animal welfare at Charles River. Members of the Pig Welfare Working Group were recruited based on interest in participating, role in the company, and area of expertise. There were 30 people involved, and they represented 16 sites within the company in 6 countries. Members of the Pig Welfare Working Group could select to participate in subcommittees based on interest, or were recommended to join subcommittees based on their expertise. There were 7 subcommittees, including behavioral management, housing, euthanasia, restraint, transportation, and specific pig care practices, blood collection, rehoming programs, and future research areas. Each subcommittee had a chair who was responsible for leading the discussion and formulating agendas. Each subcommittee also had a member of Global Animal Welfare and Training that was there to define expectations for group function and provide administrative support, such as setting meetings, taking meeting minutes, and distributing materials. Subcommittees used online meeting platforms to host their discussions and communicated and shared documents via electronic correspondence. Subcommittees met on a regular basis, 2 to 4 times monthly based on member availability, until recommendations were drafted and achieved consensus within the subcommittee. Scientific literature was used in drafting recommendations when available, but because of the lack of published peer-reviewed literature specific to laboratory pigs, select areas were comprised based on anecdotal literature and experience. The members of Global Animal Welfare and Training overseeing the Pig Welfare Working Group would meet monthly to share progress, review draft recommendations, and plan next steps. Subcommittees ran concurrently and typically met for 3 to 4 months, depending on the length of time needed to review the literature, draft recommendations, and achieve consensus. The members of the Pig Welfare Working Group met at an in-person pig refinement workshop at a U.S. site in May 2019. The workshop consisted of internal and external speakers and included a hands-on low-stress handling demonstration. At this workshop, the Pig Welfare Working Group worked together to gain consensus for ongoing recommendations. Once the Pig Welfare Working Group developed the final recommendations, The project co-chairs summarized and edited the recommendations for clarity, conciseness, and cohesion. Following approval from senior management, the recommendations were disseminated to other stakeholders. The final report was released in March 2020. The recommendations covered key areas of interest for pigs, including behavioral management, housing, handling and restraint, habituation and training, and refinements to procedures such as blood collection, study endpoints, and regular welfare assessments. Behavioral management programs should address the natural behaviors of pigs, such as providing things to chew and manipulate, opportunities to forage and exercise, and some degree of choice and control over their environment. Some recommendations address multiple areas of interest, such as social housing for pigs, which fulfills natural behaviors, but also needs to be considered when deciding on housing designs. Emphasis was put on using low-stress handling and training techniques to build positive human-animal relationships and to refine study procedures through teaching animals to cooperate. The Pig Welfare Working Group developed 12 recommendations achieved by consensus and implemented at a company level. Following rollout of the Pig Welfare Working Group recommendations, internal animal welfare assessments have occurred to benchmark where sites are and to monitor progress. Because all sites were represented within the Pig Welfare Working Group, there was 100% site buy-in when the recommendations were circulated to internal stakeholders. Sites have leveraged these recommendations to justify making changes in housing, handling, and training programs. There have been innovations, such as the invention of the piggy snuggle as seen in the picture on the left. This allows for easier and more comfortable restraint for personnel and for the pigs during study procedures. There has also been improved training and habituation of pigs to allow for blood collection from peripheral sites, such as the saphenous, radial, and ear veins. In the middle image, blood is being collected from a pig using the radial vein without restraint and following sling habituation. There has also been more emphasis on training pigs to participate voluntarily in study activities such as body weight collection. The recommendations have also been used to drive internal research for evidence-based refinements based on what works in applied settings. For example, there is sometimes pushback internally and externally when recommending additional time pre-study to prepare animals for study procedures. A simple study was designed to demonstrate highly significant and repeatable effects of basic habituation techniques on intraocular pressure in pigs, a technique in which subtle test article effects are important to define. There also continues to be collaboration and sharing amongst sites through our internal pig behavior group. This group meets 3 times per year to share recent external and internal research and present new site-level refinements. There were some challenges to overcome within the Pig Welfare Working Group. Communication was one challenge, especially with having members in different time zones and members who speak different languages. Resources and documents were shared via online platforms, and it was up to the members to keep up to date and to participate in order to communicate their opinions on the recommendations that were being developed. Another challenge was keeping the momentum going, and there was also challenges with differences in resources at each site that could affect the ability to implement the recommendations, such as money, space, time, and personnel, or differences in priorities across countries and sites. There is also a need to recognize and accept that there would be asynchronous implementation of the recommendations across sites due to these differences in resources and priorities. Maintaining focus within the subcommittees was also a challenge. The areas of interest were chosen due to need, but also due to realistic expectations of what could be accomplished. Reminding Pig Welfare Working Group members of this was important to keep the recommendations realistic and feasible for implementation. Finally, training personnel was always an ongoing challenge. The recommendations had to be communicated to the sites with pigs, keeping language barriers in mind. Personnel also had to be trained on the recommendations themselves, such as how to use low-stress handling and training techniques, and refinements to blood collection would also require site-level training of new procedures and achieving competency. In going through this process, there are reflections and recommendations we can make to anyone interested in using this approach at their own organization. First, select the topics of interest carefully. There will always be endless lists of how we want to improve welfare for animals, but selecting topics to address should be based on area of concern, what information is available for evidence-based changes, and based on what is feasible and realistic given the resources you have available. It's important to recruit diverse stakeholders in order to achieve consensus that will make implementation easy to accomplish. To allow the focus groups to operate smoothly, provide or assign administrative support. This allows the stakeholders to focus solely on sharing resources and building consensus rather than worrying about setting the meetings, taking minutes, or distributing materials. Hands-on workshops should be built into the focus group. In-person communication and demonstrations are valuable in team building and in building those lasting collaborations that will continue to move the organization forward. Build recommendations in terms of what should be done rather than what must be done. This allows choice and control over how to implement changes, and as we've mentioned, pig management programs across Charles River were very different to start with. Each site had different priorities and resources, and giving them the choice on where to start allowed sites to own the solutions and be innovative. It also guaranteed that progress was being made across all sites, even if it was asynchronous. Lastly, it's important to follow up afterwards and to reward and recognize those that are making changes. Within Charles River, the recommendations were built into our on-site welfare assessments. Sites also meet as part of the pig behavioral management calls 3 times a year to share their progress. We also have company-wide welfare awards to recognize excellence. We used what we learned for the pig welfare working group when launching the rat welfare working group, which is a much more involved project due to the large number of sites with rats and even larger number of research purposes rats are used for. Thank you for your attention during this talk. And
thank you very much, Carly, for this presentation of this very, very complex and piece of work that has involved lots and lots of people. I liked in particular the sort of the ownership that people took of the changes, the way that it was set up. And this links a little bit in with, we have a question from Hugh. You mentioned it in one of your last slides. He's asking, could the welfare outcomes of the working groups be utilized by other institutions housing research pigs? Or do you think they would need to run their own working group to get the kind of buy-in that you mentioned, which is the benefit for the focus group concept?
I think there is probably a mix there. Like I mentioned, there is a real lack of literature on how to take care of research pigs and what works best. So I think part of it is, what we learn, we will share in a publication, hopefully. But part of it is also, what's going to work at other sites based on how they manage pigs, what resources they have. And then, getting the buy-in is very important because, if you have a complex organization with multiple sites or even multiple research labs within a university, everyone's going to have their own way of dealing with those animals and working with them based on their own research needs. So I think that buy-in is very important. So that if you just go in saying you have to do this, you're going to feel like get more pushback, and unless people have a say in what's going to happen and they have a chance to give their opinion.
Yeah. Stephen is asking, were there any tensions between the subcommittees and their recommendations and the overview committee?
Not necessarily, just because the overview committee was on every subcommittee, so we were able to voice our opinions as we were drafting the recommendations. And also, I feel like, remind people that, sites might not be recommended, or, if there was conflicting recommendations being drafted up, the oversight committee could communicate that with each other and then go back to the subcommittees and kind of redraft what we're working on. So there wasn't too much tension there, as more once you go back to the sites and start laying down mandates, people might not understand why you're making those recommendations, or might make it more difficult for them as they're planning on how to address those recommendations.
You may have mentioned it in your talk, but as somebody who's worked with pigs for agricultural research and the welfare of pigs on farm, where we tend to take the husbandry methods and the environments that pigs are usually found in on a farm, also because, or not least because we want to mimic it a little bit, because that's the welfare we want to improve the situation of. To what extent could you make use of the knowledge from agricultural, farming backgrounds and agricultural research? I know you say it was both biomedical and agricultural, but nevertheless, the two are very different in their aims.
Mm-hmm. Yeah, I did my PhD research on agricultural pigs, so to me it's been interesting to see kind of what would work in a like biomedical research setting versus what works in agriculture. And I'd say, you know, especially the social research on pigs has been helpful, even though from what I've seen, the like mixing aggression isn't as common in the biomedical research pig, but yeah, type of what enrichment is more important and what bedding items or foraging items are most important. A lot of that is, of course, taken from agricultural research and what research has been done there. You know, providing straw is one of the best ways to keep pigs foraging and interactive. So a lot of that research did come from agriculture, but of course there's differences in just the environment, you know, the level of cleaning that goes on at biomedical facilities, the level of interaction they have with humans is, I think, on a much grander scale than in agriculture, where you know, they might be interacting with the human daily.
So why are there less mixing in the biomedical research? Just simply because they are kept more, I don't know, in pairs, or it's not necessary?
They're typically social housed unless there's a steady need for them not to be, but for some, yeah, even when they're mixed together, I saw some pigs, you know, come into the facility and get put into their pens, and they just didn't fight like they did in agriculture. I don't know if it's just because they're mini pigs and maybe they just don't have, like, it was in a smaller scale, so they just weren't as motivated to fight. But yeah, I didn't see the level of aggression I've seen in kind of agricultural settings. So
yeah. Thank you very much, Carly. We don't have time for any more questions, but fascinating to hear the amount of work going into trying to get this right. Thank you very much.
Yeah.
We have now come to what has become known as the speed talks. They're not supposed to be speedy, but they are short. We are going to hear 4 short presentations lasting 3 minutes each. So if you do the math, that will be 12 minutes. If you have questions, so first of all, hold on to your hats because it comes very, very fast. If you have questions for these talks, please put them in the question box, and can you please write the name of the presenter in front of it, so that at the end of this session, when the 4 presenters come in for their questions and answers, we can know which question is going to be asked to whom. So with no further ado, I would like for Stephen to put on the 4 short presentations, please.
Hi, my name is Fiona Dale, and today I'm going to talk to you about changes in separation-related behaviour in Generation Pup puppies aged from 12 weeks to 6 months of age. So separation-related behaviours are a welfare problem which occur when owners are absent and typically occur within the first few minutes of owner departure. And these include destructive behaviours, excessive vocalisations, as well as other signs such as lip licking and depression-like states. And depending on the paper that you're looking at, the prevalence has been put anywhere from 6 to 55%. Not a lot of work has looked at how these behaviours change over time. However, one paper has suggested that they stay relatively consistent over a 3-month time period within the first year of them being in a new home. And understanding how separation-related behaviours change over time could help with time-specific targeted behavioural interventions. So with this in mind, we used a longitudinal study to further understand whether and how these behaviours change from 12 weeks to 6 months of age. We used a Dogs Trust longitudinal study called Generation Pup, where owners sign up to answer questionnaires throughout their dog's lives on their dog's behaviour and health. We used the 12 weeks and 6 months questionnaires, and our binary separation related behaviours of interest were barking, howling, whining, chewing items other than toys, and scratching at furnishings or doors when puppies were left alone. And we used general estimating equations to look at the effective times of time on these behaviours. We had data for 284 dogs. At 12 weeks, 35% of dogs showed 1 or more separation-related behaviours when they were left alone, compared with 47% at 6 months of age. This may be not what we expect, as we may expect that these behaviours will go down in frequency over time as puppies settle into their new homes and get used to being left alone. We found a weak but significant positive correlation between the numbers of separation-related behaviours dogs displayed at 12 weeks and at 6 months, suggesting a similar pattern at these 2 time points, and potentially that these behaviours are consistent, or dogs are consistent in the way that they respond to being left alone. Looking at the behaviours themselves, we found that whining significantly decreased over time, whereas barking, chewing of items other than toys, and scratching at doors and furnitures significantly increased over time, whereas howling was infrequently reported. What's going on? It may be that whining dogs are generally getting more comfortable being left alone, or that they're showing anxiety about being left alone in a more subtle way. The increase in chewing of items or scratching of furnishing or doors may reflect changes in activity or teething that's happening over this time period. And the increase in barking may be that these dogs are responding to external sounds, or potentially that they are responding in a way that has successfully brought their owners back to them and are continuing to act in that way. So changes in separation-related behaviours reported by owners were observed between puppies aged 12 weeks and 6 months of age. And we have to keep in mind the influence of owner report and owner perception, as these may affect these results. And further work is needed to investigate the impact that factors such as time left alone, activity levels and teething have on these behaviours. And thank you for listening.
Hello, my name is Sara of Owczarczak-Garstecka and I'm a researcher at Owczarczak-Garstecka. Data for the study come from the Generation Pup project, an ongoing longitudinal study of dogs aiming to recruit 10,000 dogs. Generation Pup aims to report incidents of health and behavioral problems, identify risk factors for health and behavioral outcomes at different stages of dogs' lives, and to inform preventative strategies designed to improve dog welfare. Surgical neutering is the most common surgical procedure for dogs in the UK. Whilst the best timing to neuter a dog depends largely on their size, sex and breed, impact of timing of neutering on healing is unknown. Therefore, this study aims to report on common post-operative care after neutering and wound healing problems, and to compare presence or absence of owner-reported wound healing problems between sexes and dogs neutered at 7 and 15 months. Generation Pup data are collected through regular owner-completed surveys and using other tools of data collection. For this study, data from the 7th and 15-month surveys were extracted. In particular, we were interested in questions on dog surgical neutering, use of protective clothing after neutering, and how the wound healed. These differences were compared between the 2 time points and between sexes with chi-square tests. At 7 and 15 months respectively, 13.3 and 43.5% of dogs were castrated, and 19.5 and 46% were spayed. Significant differences between sexes in the type of protective clothing worn were identified at both 7 and 15 months. Post-surgery, on average, proportionally more male dogs wore an Elizabethan or busted collars, but more female dogs wore a t-shirt or nothing. No difference in the type of protective clothing worn between age groups were found. At both 7 and 15 months, less than a third of dogs experienced healing problems. Only at 7 months the difference in healing problems between sexes was significant, with proportionally more female dogs experiencing issues, possibly due to spay surgery being more invasive than surgical castration. The difference in healing between age groups approached statistical significance for male but not female dogs, with more male dogs experiencing wound healing problems at 15 compared to 7 months. This study identified significant differences in post-operative care and healing between sexes. In our sample, a quarter of dogs were reported by their owners to experience problems with wound healing post-neutering surgery, suggesting further support to owners regarding post-operative management could be beneficial. I'd like to thank our funders for supporting this project, dog owners who completed the surveys, and veterinary and other professionals who continue to help us recruit participants. I'm also grateful to the wonderful Generation Pup team for their ongoing support.
Hi, I'm Martin van den Broek from Ghent University, doing a short talk on age estimation of puppies in cross-border trade. In the European Union, puppies younger than 15 weeks are not allowed to cross borders. This is imposed by EU rules that were designed to protect the health and welfare of the puppies and to prevent the spread of zoonotic diseases such as rabies. It is said, however, that underage puppies with falsified passports are being traded. To combat this, a better law enforcement is crucial. Therefore, the aim of our study was to assess whether the legal age mentioned on their passports of puppies that were imported in Belgium corresponded to their actual age, estimated based on the age characteristics that they displayed. Also, we wanted to find out which age characteristics are suitable for age estimation in forensic setting. We evaluated the eruption status of the dentition of 10 Golden Retriever and 10 Pomeranian puppies aged between 119 and 129 days, that is 17 weeks and 18 weeks 3 days, that were imported from 3 Eastern European countries and were advertised for further sale by 4 breeder merchants in the region of Flanders in Belgium. We did the same with the carcasses of 6 Pomeranian puppies that came from 4 Eastern European countries and that died at an age between 112 and 127 days. Of these carcasses, we also took radiographs to evaluate the formation of ossification centers in the carpal region and in the elbow region. The outcome for each puppy was then compared to a reference interval that we established based on literature and breed-specific results from locally bred puppies. In 6 Golden Retriever puppies, the passport age exceeded the dental age interval, demonstrating that these puppies were younger. Old Pomeranians still had their deciduous dentition, placing them in a wide dental age interval which could have masked age fraud. However, based on the radiographs, it was found that the passport age exceeded the bone age in 5 of 6 of the carcasses. Conclusion: it was undeniably demonstrated that a considerable part of the puppies in this limited study were actually younger than mentioned on the passports. Age assessment in a forensic setting is possible, but for this age category it is best to combine dental age and bone age. And of course, more breed-specific data and data on intermediate stages are necessary. Thank you.
Hello, my name is Guillermina Hernandez Cruz. I'm a PhD student in the Animal Welfare and Behavior Group of the University of Bristol, and my talk today is entitled Behavioral Diversity of Captured Monkeys in Rehabilitation. Behavioral diversity indexes are used to investigate the effect of certain behavioral practices, such as environmental enrichment. In many cases, these practices are aimed at improving welfare of the animals. In this study, we used the Shannon Age Index to assess the rehabilitation outcomes of 16 captured monkeys that were rescued from the illegal trade in Brazil and were being kept in captivity before release, so before reintroduction, in a rescue center in northeast Brazil. We hypothesized there would be an increase in behavioral diversity when comparing the start and the end of the 4-month rehabilitation program. We performed 47.5 hours of behavioral observation and calculated the behavioral diversity indexes for the baseline and final phases, and then we calculated the difference between them. This was called F-V scores. We performed one sample t-test to see or to test if the F-V scores were significantly different from 0, which they were. Moreover, a two-way analysis of variance showed males had significantly higher F-V scores than females. This means that males increased more their behavioral diversity, significantly more when compared to females. But overall, all animals significantly increased their behavioral diversity, as evidenced by an increase in the behavioral diversity indexes. We didn't find any significant difference related to age. We conclude that the rehabilitation seems to be helping the animals to increase their behavioral diversity. Behavioral diversity indexes may help in the behavioral assessment and welfare monitoring of rescued primates, but it is obviously very important to consider that they need to be used in the presence of other indicators. This could be physiological indicators such as cortisol, or other behavioral indicators such as activity budgets, or ideally a pre-release and then a post-release monitoring. Thank you very much.
Thank you very much for these 4 fast talks, and I would like to invite the presenters to turn their camera and microphones on, and we will see if we can. There we go. Sara, Fiona, and Kilomina, are you there as well? Yes. Excellent. Thank you very much.
The camera is not working yet.
Yes, I think you're all good. We have some questions. I'll try to see if not necessarily in order, but I will start with a question for Fiona. Do you think that the older dogs were showing separation related behaviors? Because, sorry, do you think that the older dogs that were showing these behaviors are changing their behavior because they have found better coping behavior to deal with separation?
Yeah, potentially. Yeah, that's quite a good point. I guess also this is based on owner report as well. So we're potentially seeing a change in how owners are perceiving these behaviors as well, and what they're seeing as being acceptable as well. But that makes sense. It's over a 3-month period, so it might be that they found, you know, if they're barking a lot or where they're doing whatever, it's not being responded to in any way. So they're changing their response to being left alone accordingly. But yeah.
But was it, just an additional question, was it necessarily the same dogs that were doing the same behaviors at 3 and at 6 months? I mean, I know there were changes, but if there was a reduction in whining, that doesn't necessarily mean that all the dogs that kept on whining also had whined at a younger age.
Yeah, it's quite difficult to track them completely. And so like dog to dog, see, that's a really difficult thing, that we were trying to work out a way that we'd be able to do that. I did have a look at characterizing whether, it's a weird way of doing it, like looking at whether dogs always did something and stopped, started, or never did it. And yeah, so that's something that I'm kind of trying to tease out of the data. So yeah.
I have a question for Sara. This is about the different types of clothing that the 2 sexes are being dressed in. I mean, is that only due to the position of the wounds, or is there a tendency for female dogs to be given a different dressing than male dogs for other reasons?
That's a good question.
You're allowed to speculate.
That's a good question. I don't have an answer to it really. My guess is that some of the difference in clothing might be due to position of the wound and how easy it is for a dog to access the wound, to lick it or scratch it, if, you know, if they are suffering from pain or itchiness after the surgery. So it could be explained with that.
Also, you mentioned that a quarter were experiencing post-surgery issues. That seems very high. Why is this, and is this acceptable? I know it's not your responsibility, but what can we do to avoid this?
So because it was a very short presentation, we kind of put all of the issues together. So the range of those issues was quite varied, and some of these included infections and potentially quite serious issues, whereas others was just an owner reporting that the dog was itchy, or the dog was scratching it, or that the wound was kind of lumpy afterwards, which might have been a normal process of healing. Again, we relied on owner reports. So you know, if all the dogs were experiencing very serious issues with healing, that definitely would not be acceptable. But because we are just summarizing very kind of preliminary data, this could include a whole range of issues, and some of it may be just an owner's perception of what's not to them normal way of healing.
Yeah. Thank you. I have a question for 2019. Interesting, all these forensics and puppies. I'm just wondering, these can mostly, well, they can, you have done them post-mortem. Can they be used on live dogs, or are there different ways we can sort of identify these very young dogs before?
Yeah, well, the study existed from two parts. So there was one part that we did, there was a longitudinal study in which we followed up the teeth exchange in puppies to establish reference data, and that was in living dogs. So that is something that you can do in living dogs and living puppies, and just check the teeth. And then taking X-rays is also possible in living puppies, but of course they have to be sedated for that. So therefore, for practical use, like for forensics or legal purposes, you can do that. But of course, for the study, we didn't do that in living puppies. We used puppies that died because we didn't want to expose living puppies just for the study to X-rays. So, but yeah, and practices both methods are used, can be used. Yeah.
Okay, thank you. Guillermina, there's a question here for you. How was the BDI recorded in relation to enrichment events? Were the behaviors observed during the enrichment, or were they observed all the time?
No, the behaviors were observed all the time. So the behavioral diversity is calculated based on a general activity budget. We didn't really, I mean, there were environmental enrichment in the enclosures of the animals, but it wasn't part of the experimental procedure to actually measure responses to environmental enrichment at all. So they were there, and that was all. Really, the point was seeing how the animals came from being, kept as pets. These are monkeys that are rescued from the illegal pet trade, and then they become used to being with other monkeys in captivity for them to be ready to be released. So it's kind of just going with the general procedures of the rescue center and seeing if only that is enough to actually increase the behavioral diversity. So this was kind of a study to see what specific environmental enrichment we could do in the future. But we didn't do any environmental enrichment as part of this study, no.
Okay. I'm afraid we are out of time. Thank you very much for not only presenting your work in such a short time, but to answer the questions that have come in. It's very enjoyable, and thank you very much.
Thank you.
Thank you.
I will now present our next speaker, which is another early career researcher of the year winner from UFAW. This is Dr. John Chou, and not Chen. And she's going to talk to us about, if you judge a pig by the tail, how a solution-driven approach can open up new possibilities to animal behavior and welfare research.
Hi everyone, I'm Chou. I'm currently a postdoctoral researcher at the Swine Teaching and Research Center at the University of Pennsylvania. I would like to thank UFAW for the recognition of my work. It is an honor to receive the Early Career Researcher Award alongside Dr. Jamie Dallaire. It is a huge encouragement, and I would like to thank my supervisors and mentors during my PhD year, Dr. Keeling O'Driscoll from Chagas, Dr. Reed Dees and Dr. Dale Sandecrock from SRUC, Dr. Irene Kamerling from the Polish Academy of Sciences, and Professor Alistair Lawrence and Professor Natalie Warren. They have given me support, encouragement, and guidance, and also inspirations during my PhD years. Thank you for your confidence in me. Today, I'm really happy to have this opportunity to talk to you about what I did during my PhD and a little bit about my current project, and share with you some of my personal reflections along the way. First, quickly about myself. I come from a social science background. I did a MA in Science, Technology, and Society in Taiwan, and later did the MSc in Applied Animal Behavior and Animal Welfare in Edinburgh. So both disciplines are very multidisciplinary, so my training has always been teaching me to understand the complexity of things and to be able to see things from different perspectives. But because I have experience with animal welfare groups, when you deal with real life animal welfare issues, you really want answers, you want to take actions. That's why I am very drawn to a problem solving approach, and also why I did my PhD project, which is trying to identify strategies to manage tail biting in pigs housed in fully sedated systems, and also my current postdoc project, which is trying to find the best way to manage post-swing cells. Both projects are really trying to deal with very specific issues. Although my PhD project is about pig and tail biting, it actually all began with wood. So at the time when I started my PhD, my colleague Amy Haig and my supervisor King O'Driscoll, they did a survey on Irish farmers, and they asked them what do they think about environmental enrichment. So the farmers are interested in wood, but they have their concern. They're not sure whether wood is safe for the pigs to use or whether the pigs actually use them at all. So we use this as a starting point to have some conversation with them about wood, about enrichment, about tail biting, and eventually about tail docking. So I probably spend more time measuring all the different wood types than I measured pig in the first year of my PhD. But what we found was that using wood is not as simple as we thought. The different wood types actually mattered, and the pigs like to interact with softer but not necessarily the wood type that had a higher moisture level. And during our two studies, one on the commercial farm and one on our research farm, we didn't actually find any major effect on tail biting or tail lesion. But overall, we did have low level of tail biting because we were working with docked pigs at that time. And we did confirm that wood posts are safe for the pigs to use. However, this has become a foundation for our work later on. So our next logical step was to really start working with undocked pigs. So in this study, we selected the enrichment items that were identified in the previous two studies that the pigs interacted with the most. And then we try to keep everything similar to what's happening on the commercial farm in terms of stocking density and management practices. Another factor we investigated was the dietary fiber, and this is also another thing that the farmers identify that they are interested in, and it's easier to change in terms of their management. So what we found was that unfortunately higher fiber diet actually caused more tail biting in our study, and the enrichment items, although they were interactive with by the pigs, they had very marginal effect on tail biting and tail lesion, and overall we had quite a high level of tail biting. So we now know that if we want to manage tail biting in undocked pigs on fully sedated floors, we have to do more than just altering the fiber in their diet, and we have to give them more enrichment. So in the final stage of my PhD, we really try to explore all possible ways to provide meaningful enrichment that is still compatible with fully sedated floors. So we did that by conducting a pilot study first, in which we just tested out so many different items, as you can see here on the slide. And in the final study in my PhD, we tested whether given the enrichment pre-weaning or not have an effect, and also coupled with post-weaning, so different enrichment replenishment strategies. So basically, we try to find a balance between giving the pigs the most access and what may be feasible on the commercial farm. So what we found was that the tail biting level was indeed lower compared to the previous study, but the pre-weaning enrichment didn't have much effect on tail biting and tail lesion. And the most frequent enrichment replenishment, which means that the pigs had almost ad libitum access to all the different items, they perform the least harmful behaviors, and it also promoted their growth. So this last finding came as a surprise for us as well, considering that actually all our pigs had access to multiple items. The only thing different was that we replenished the enrichment at different schedule when they were depleted. So at the end of my PhD, at least we can tell the farmer that we have meaningful way to provide enrichment to undocked pigs on fully slatted floors. And then, of course, there will be costs and additional costs involved, but then it could potentially be offset because you will get a profit margin, better profit margin because of the benefit the enrichment can bring. At the same time, when we were working with undocked pigs, we designed a tail biting outbreak control protocol because we expected that there will be a certain level of tail biting. Ethically, we know that we need to intervene when something like this happened. And it also allowed us to test out the protocol within our main studies and without needing to use more animals, so that we can actually provide a really useful recommendation for the farmers to deal with tail biting outbreaks. So we have these 3 different methods, and we randomized them, and then we tested out during our main studies. And we were able to actually control 80% of the severe tail biting outbreaks because our definition of outbreak, the threshold, was higher. And we are also able to reintroduce all the pigs if they were removed from the pens back to their home pens. And we think that this is a very important point because on a normal farm, you won't have unlimited space to keep removing the animals. During my investigation on a very specific subject on tail biting, new ideas also emerged. So as I was watching all the time the pig's tail from when they were docked and later on they were not docked, and you started to really appreciate how expressive their tails can be. And I noticed that for individual pigs, they actually will curl their tail on different sides. So I started to follow the really nice work by Charlotte Gusso and her team on tail lateralization. And at that time, still not many people actually look into lateralization in farm animals. So we want to see what tail biting, what role tail biting can play in terms of pigs' tail lateralization. So our initial finding was that our pigs do also have a tail bias, or whether they like to curl to the left or curl to the right. And when they were recorded a higher tail damage score, this default tail bias becomes less consistent. But we don't know whether it's because they're feeling discomfort or pain, so that's why they're changing their tail bias more, or is it underlying mechanism related to their emotional state. But we think that it will be really interesting to explore further this aspect. At the same time, I also started to ask different questions beyond just tail biting and normal husbandry practices, because when I see firsthand how tail biting was affecting the pigs, I started to wonder how their experience was as a tail biter, a tail bitten victim, or neutral pig that was not involved in tail biting. So I tested these 3 different tail biting phenotypes in a novel object test at 2 different time points. Once when the tail biting was still quite bad, and then later on when tail biting comes down. So what we found was that they did respond differently to the novel object test, but then we didn't find any evidence of any long-term consistency between these 3 phenotypes, which may mean that the experience of tail biting may not as long-lived as we expected. But I think asking these questions really urged me to understand more about their psychological experience and the psychological impact of tail biting beyond just tail lesions and productivity. My current postdoc project is trying to find ways to best house post-wing sows. This project is also to answer a very specific question posed by a recent legislation in California in the United States on almost zero confinement of breeding animals. So the post-weaning period is a sensitive time for the sows because they psychologically they are going through the separation stress from their piglet, and physically they are also in a very delicate state because they lost a lot of weight after lactation. And in EU, in some countries, sows are also housed in confinement stall during this time period. And I first did a very thorough literature review trying to find out what we know so far about how to house them during this time. It is still inconclusive, and very few people actually ask the question of what is sow psychological well-being and their subjective experience during this time. So I designed a modified novel arena test. We expose them either to a novel object or the stall that they will be housed in. And we test them before they were housed in the confinement stall and a week afterwards and see how they respond to the stall differently. I'm still currently processing all the data, but this is just preliminary data and all descriptive. But judging by the current data, I think it kind of showed that there seems to be a difference in the time they spend in the stall, especially for the sows that were actually housed in stall for a week. So you can see that they reduce more time they spend in the stall compared to the other groups. And just during the brainstorming of planning for this project, we really constantly ask about how do we evaluate the subjective experience of the animal, and really urge us to go back to the literature and to find out more question and answers about the most fundamental part behind these questions. Due to the constraint of time, I couldn't really go too much into details about each of the study. If you're interested, all the study I did during my PhD have been published, and please feel free to ask me questions later or check out our publications. So going back to the title of my talk, I've been thinking about my career as an early career scientist and always taking this very solution-driven approach. And there have been some discussion that this type of approach can bring some limitation to what we can achieve. I hope that I have demonstrated that in my case, I don't really feel that it has given me any limitation on how to ask new questions or how to answer questions in new ways. For me, the fundamental and the applied always goes hand in hand, and the only restraint we have is probably the time and resource we have, which may probably be solved by more collaborations or conversations or being really open-minded about the complexity of things. For me, it's also very important to start from a small thing, like I did with my PhD. I started with trying to understand everything about wood, and then later move on to bigger and bigger issues. I think it is really important to build out an investigation like this, and then it also helps us to open up conversations with different stakeholders. And another thing I find it very important is the time I actually spend with my pigs. And just by watching them during the day-to-day care or during the data collection time when I was observing them, or just by talking to the technician or staff which are on the ground every day, it always gives me more ideas or inspirations to tackle the issues I have at hand. And there is more and more research on human-animal relationship, how that will help the animals or help better stockmanship. I think for animal welfare scientists, it is equally important. For example, I found a very special pig, as you can see here, number 22. I think it is a good reminder for me to keep thinking about their individuality, about their uniqueness, so that I won't stop asking the more difficult questions. And finally, just I think it is really important to keep thinking back on what I have done and then what I'm doing now, and always keep at the back of my mind the more difficult ethics question and about why I started to do animal welfare science in the first place. I sometimes got asked about, because I'm working on an issue so specific, the fully static systems, does it mean that I'm happy with the system? I think just because I'm working on finding solutions to a very specific issue means that I will just stop here and I won't ask more questions. I believe in baby steps, so I think taking a step-by-step measure, it can make future changes possible. Finally, I just want to acknowledge again my wonderful supervisors and mentors, and also the research staff and my colleagues I have the pleasure to work with. They have been tremendous help and also inspirations for me during my PhD, and also my current postdoc team, Dr. Tom Parsons and the swine unit staff at the new Bolton Center. I just want to leave you with kind of a final message. I think a pig's tail is more than a tail. It is really my gateway into the world of applied animal behavior and welfare science without limits. Thank you for your attention. I'm really happy to answer any question you may have.
Thank you very much, Jen. That was fascinating. And there you are in real life. Welcome. Yes, lots of questions. The first question is from Jamie, who is saying, I may have missed this, but was the decreased lateralization of tail curling in bitten pigs something that occurred only after their tails were bitten, or also before? And could it be related to how predisposed individuals are to being targeted and bitten?
Yeah, that's an interesting question. Thanks, Jamie. I think for the results I just showed, it is when I record the score. So basically that means that it is after they were bitten. So that's why I was saying that this could be related to because their tails were bitten. So they've been changing the lateralization more. But then I think, yeah, that it will be interesting, right? I mean, to understand what is causing the lateralization in the brain, and then maybe something already happened even before their tails were bitten. Thanks.
Thank you. The other question comes from Jean-Luc, who asks, well, he says, very interesting approach, Jen. When you mention the solution-driven approach, in your opinion, how far should we think about the solution? For example, we know that fully slatted floors are a major welfare risk to pigs. So should we still be trying to make it work, or should we raise the standard and compare it to a better system like partially slatted only, even though the solution may sound difficult at present to some in the farming community?
Yes, thanks, Jean-Luc. Yeah, I think that's a difficult question, right? I think this is, yeah, that is also another question I was mentioning at the end of my talk. I constantly get asked, but I think, yeah, so I was mentioning that I do believe in a step-by-step approach. I mean, before we actually tried out my studies, we weren't sure that we can ever find any solution that may work on fully static systems. But I think the fact that we did find something that will work, I think that is kind of a starting point to start asking the farmers to do something. Okay, so now you can start improving even within your system, but then gradually, of course, we will like to move on, to polyslatted floor and even maybe to, outdoor access, concrete floor and things like that. So I think that's why I was saying that, this solution-driven approach shouldn't be the end of your investigation. It should only be the starting point. And then we don't necessarily will find solution on all the, the subject where we're trying to investigate, but then it is a starting point for me, definitely.
And it was interesting as well, because you said that you found that you actually found some improved growth in some of these pigs that had very frequently changed enrichment, and that would improve the profit margin and be a great incentive. But the downside, the flip side, I suppose, is that changing those enrichments so frequently is labor intensive. Would you like to comment on that?
Yes, I think that is the part that I mean, we didn't manage to do during my PhD. In our paper, we say that, yeah, it is very important, the labor part. So that's why in my last study, we actually tried to see that, you know, different replenishment strategies. So let's say the most labor intensive, you just have to constantly replenish them, and to the least one, let's say replenishment 3 times per week. How do we find a balance between those strategies? But of course, I mean, the more we can give to the animals, the better they will perform. I mean, at the end of the day, we're trying to find really a balance between how much labor you can really put in to replenish your enrichment, and how much more we can give to the pigs. But yeah, I think the labor part is definitely should be accounted for.
And the fact that, well, by using enrichment and different techniques, we can try and prevent outbreaks of tail biting, and when it happens, we need to spot it quickly and remove the pigs. You did show a number of pictures and mentioned a couple of times pigs that had been, you call them treated tail-bitten pigs. How do we treat tail bites, and how efficient is that?
So our basically our tail biting outbreak protocol. So I did say that our protocol has a higher threshold. So basically, which means that the tail biting has been already bad, and then we started to intervene. So when we use, of course, we need to use some antibiotics, and then also pain reliever to help them, and then sometimes we remove the victims so that they can stay outside the pen for maybe up to 72 hours before. If they recover, then we reintroduce them, which actually works. I mean, we managed to actually reintroduce them back. We always just remove them in pairs or in groups, so that we make sure that when they go back, they're not going back alone. So by this type of medical treatment coupled with some animal movement at the time of tail biting, we manage to control the tail biting outbreaks.
Okay, interesting, very interesting. Well, I'm afraid time is running. Thank you very much, Jen, for enlightening us on this. Congratulations again on your prize. And thank you very much. We will now go to the last talk of this whole conference, and the honor of giving that goes to Raúl Guevara, who will be talking about the systematic review of animal-based indicators to measure thermal, social, and immune-related stress in pigs.
Hi, good afternoon. Thank you so much for being here today. My name is Raúl David Guevara. I am a first-year PhD student at the Autonomous University of Barcelona, and this is the first part of my research about stress indicators in pigs. And the name of this presentation is Systematic Review of Animal-Based Indicators to Measure the Thermal, Social, and Immune-Related Stress in Pigs. So first of all, some definitions to keep in mind during the presentation. The first one is stress is the group of physiological responses to recover the balance of the organism. Stressor is any factor that can reduce the animal welfare. The animal-based indicators is the measurement of the animal responses to a certain stimulus, and the indicator should be valid, reliable, and feasible to generate proper conclusions. Finally, the animal-based indicators are obtained in a precise way and usually are presented in a quantitative way. Therefore, the intensification of pigs production has increased pigs' exposition to stressful factors such as extreme temperatures, which produce thermal stress, high social density or negative social interaction, which produce social stress, and allergens, pathogens, and other microorganisms that may challenge the immune system of the animals, provoking immune-related stress. Then the objective of this literature review is to offer an insight into the variety of indicators and methodologies commonly used to evaluate the effects of different stressors in the physiological response of other animals. The systematic search review was performed following the PRISMA systematic search guidelines in 3 search engines: ScienceDirect and Scopus from Elsevier, and PubMed from the National Center of Biotechnology Information. Peer review literature from 2000 until 2020 in English was considered for the literature review. On the other hand, literature review manuscripts, in vitro studies, transport stress studies, social isolation, new environment, or reproduction studies were not taken in account. Only peak studies were taken in account for the literature review. The search literature was performed by one researcher, and the search results were validated by the other 2 researchers. Now, the search terms were selected trying to answer the research question: which are the animal-based indicators to measure the impact of the stress from different sources, thermal, social, and immune-related in pigs? Then, each stress model has 3 search terms: animal, stress, and indicator, and the literature searches were the combination of one word from each group. So the query line will be something like this. The search terms were searched in the title, abstract, and keyword sections, and the stress indicator, the methodology, and the author's information were extracted. Additionally, for the immune-related stress model, the immune challenge was recorded. Now, the results of the thermal stress model: 144 articles were retrieved from more than 3,000 results. Duplicate articles and articles that didn't fit the selection criteria were discarded, and the articles were grouped by the indicator measurement. It is important to point that the articles could fit in more than one indicator, as the studies measure several variables. So 7 indicators were identified and 39 methodologies were used. So here, the body temperature was the main indicator measure, especially through rectal temperature. The respiration rate was the second indicator, and it was assessed mainly by the count of the flat movements of the animal. And the physiological marker were mainly assessed through blood samples, and particularly the concentration of the glucose in the blood. Other indicators were the skin temperature, which was mainly measured with non-contact infrared thermography, environmental indexes, which was assessed with the temperature and humidity index. The lying behavior was mainly detected with the direct observation of the behavior of the animals, either through scan samples or video review classification. And the less applied indicator were the vocalizations through the sounds feature analysis. In the social stress model, 197 articles were reviewed from more than 2,400 search results. Articles were grouped by the indicator measurement in the studies, and 49 methodologies were identified. For this model, the indicator of social interactions was the most measured, and mainly through direct observations, particularly the fighting behavior, aggressive behavior, and the biting. The physiological markers were mainly assessed in blood samples, and especially the cortisol. Lesion score was the main methodology used to assess the body lesions. The weight gain of the animals was the methodology most applied to measure the animal performance of the animals. and the vocalizations of the animals was the methodology less used in this model. In the immune-related stress model, 535 articles were retrieved from more than 32,000 search results, and the articles were grouped by the indicator type. Here, 82 indicators were identified, and the blood chemistry indicators kind was the main indicators measured for this model. The cytokines activity, either interferons, interleukins, or tumor necrosis factor were assessed the most in this review. The detection of the clinical symptoms was the most used methodology to measure the physiological markers. The animal weight gain was the main methodology to measure the animal performance of the animals. And the direct observation was the methodology most applied to assess the behavior of the animals, and particularly the detection of the depression behavior. Additionally, for this model, the immune challenge was recorded, and the main challenges used by the researchers were the LPS from the E. coli, the porcine reproductive and respiratory syndrome infections, and the salmonellosis infection. So, based on the screening of the indicators during the literature review, we categorize these indicators in 3 groups based on their relationship with the stress. So, the causal indicators are the ones that measure the magnitude of the stress factor. The biological response indicators are the ones that assess the physiological response triggered by the stress factor. And the consequence indicators are the ones that measure the production consequence caused by the stress factor. Additionally, the indicators were evaluated by their validity, reliability, and feasibility to suggest a proper methodology to measure the indicator. For the thermal stress model, these are the general physiological reactions. There is a reduction of the feed intake, an increase of the respiratory rate, water intake, and the peripheral blood flow, and a higher incidence of the lying behavior. Now, the environmental indexes were presented as causal indicators, and the methodology suggested to measure this indicator is the temperature and humidity index. The body temperature, skin temperature, respiration rate, and physiological markers are presented as biological response indicators, and the rectal temperature, non-contact infrared thermography, the count of the flanks movements, and the assessment of the glucose concentration are the methodology suggested to measure these indicators. Finally, the behavior and the animal vocalizations were considered as the consequence indicators. The behavior is suggested to measure through direct observation, either through scan samples or video review assessments. And the animal vocalizations is an indicator that is rising among the behavior and welfare researchers. And so far, vocalization of hunger, thirsty, pain, and fear have been identified through the study of the sounds features such as intensity, frequency, wave amplitude, among others. In the social stress model, these are the general reactions. There are alterations of the blood metabolites concentration, like the cortisol, acute phase proteins, and the immunoglobulins. The animal performance is affected, and the immune system is activated. So social interactions and lesion assessments are presented as causal indicators, and the methodology suggested to measure these indicators are the direct observations and the lesion score. Physiological markers are presented as biological response indicators, and the suggested methodology is the measurement of the cortisol either in blood or saliva. Consequence indicators are the animal performance and the animal vocalization, and the animal performance is affected because of the biological response, and mainly can be measured through the weight gain, and the animal vocalization features were mentioned before. In the last stress model, these are the general reactions to immune challenges. So there is an activation of the pro-inflammatory cytokines, there is expression of clinical symptoms, and a reduction of the animal performance. So during the immune challenges studies, the nature of the immune challenge is previously known, so there is no need to identify it or measure it again. However, there are analytical tests to verify the presence of the virus in the organism, and those tests were grouped within the physiological markers. So the blood chemistry and physiological markers were the indicators presented in the biological response indicators, and the cytokines activity and clinical symptoms detection were the methodologies more applied and are the ones suggested to measure these indicators. Finally, the animal performance and the animal behavior are the consequence indicators, and the methodology suggested are the evaluation of the weight gain of the animals and the direct observation of the behavior, especially to detect the depression of or the sickness behavior. So to conclude my presentation, I want to state some take-home messages. The first one is, after the assessment of validity, reliability, and feasibility performed in the literature review, it was possible to observe that biological response indicators were more sensitive to the stress detection. Then these indicators should be used to detect the stress factors and to take decisions toward actions that reduce poor animal welfare. Also, it was possible to observe that the methodologies were invasive, less invasive, or non-invasive. So future endeavors should focus on the development of non-invasive methodologies to ensure constant monitoring, more accurate measurements, and the reduction of the stress animals caused by the handling needed to take the measurements. Conclusively, no indicators have been found that may provide insight into the welfare of the animal regardless of the stress factor. So future research in this area is needed, and also there is a need for specific indicators that may help to identify the nature of the stressor to be able to take decisions toward better animal welfare conditions. So thank you for your time and your attention, and I am happy to answer your inquiries.
Thank you very much, Raúl. Very interesting and a big piece of work. We have questions coming in for you. First of all, why did you decide only to use the last 20 years of literature?
Well, we wanted to take the more updated information. It was like kind of just pointing. We just decided to take those ones because the idea was to take the more updated information mainly.
Okay, but there could be other things happening in the past. I'm saying that because 20 years ago, I did my PhD more than 20 years ago, so I'm sort of feeling, really, is that how we look at it? But okay.
Yeah. Well, it also in the last 20 years, like there have been a great increase of the technology, like machine learning vision and detection of movements. So we wanted to capture that information, like what is the technology or what are the sensors that they are using to measure these variables.
It was interesting to see because I sort of expected there to be a lot of ways to measure immune-related things, and there was. You found 82 different methodologies, but I was somehow surprised that there were so many different ways to measure both social and thermal stress. You mentioned 49, 39 different ways. Did that surprise you?
Yeah, that was one of the scores that we found, because like as you can see, like in the first model, in the thermal stress model, like things are more clear. Like you see that which are the causal indicators, which one are the biological response indicators, are which ones are the consequences. And because of the design of the model, like the immunologic, the things were kind of more dispersed, and mainly because all the measurements have to be made in the animal directly, so there are not way to take the measurements in a remotely way. You know what I mean? So yeah, it was kind of one of the scores that we found.
Yeah. I like the way you presented it, or your division of things into the causal, the response, and the consequences that the measurements were reflecting. I thought that was quite interesting. But does that tell us something about how the measurements, the validity of the measurements? I mean, the old chestnut is cortisol for social stress. We know that cortisol goes up whether you have an excitement, whether that excitement is positive or negative. What does it say about the validity of the different methods?
So mainly we try to find like which are the main indicators, and then related with the stress relationship. So it was kind of like that. Like I tried to answer the question.
It's a difficult question, though, so don't feel you have to be able to. I was just wondering whether you had found that some were better predictors of social stress or thermal stress than others, but maybe that was not also the aim.
Yeah, like the main objective was like to quantify and to see like which are the trends, like what are the most popular indicators, and at the end also based on the terms of validity, reliability, and feasibility, we wanted to recommend like, hey, if you have an experiment, we suggest you to measure this and this and this, because these ones are the more popular, but also like these ones are the more valid, more reliable, and more feasible to practice in the experiments.
Yeah, sure. Thank you very much, Raúl. It's never easy to be the last presenter of a Congress. You did very well. Thank you very much for being here.
Thank you so much for the opportunity that you gave me to share my work with you. Thanks for all the audience for staying here until this point, and I hope to see you next year, or maybe presently.
That would be lovely. Thank you very much.
Thank you so much.
And now we have come to the end of the Congress. I will hand over to the CEO of UFAW to close down the Congress officially. Thank you very much. Hugh.
Thank you, Birte. I am going to share one last PowerPoint with everyone. Hopefully you can see my screen. So yes, thank you everyone for a wonderful meeting. It's been an absolutely fascinating couple of days. I'm very inspired by how much really exciting, innovative animal welfare science is going on, and I hope you were too. So just again, a couple of points at the end of this meeting. If you're still watching, we'd really love you to support us to keep doing these meetings and keep making them free. And we'd really love you to join UFAW, because that both helps us do that and will be a really good thing for you if you're an animal welfare scientist, because we really should be the society that you're a member of, because it's really our job to support what you do. But you can also donate if you'd like to support the meeting, and thank you very much for those of you that did. If you're wondering where to publish all that fascinating animal welfare science that you've just told us about, do have a think about our journal, Animal Welfare. It's a really good home for animal welfare research. You'll have a really fabulous editorial experience with our editorial team, who are all qualified vets or animal welfare scientists who can do a really great job of shepherding your paper into publication. So do consider animal welfare when you're publishing your research. Do make use of the discount code for the Wiley book series. You can use VIII 20 or the one that's in your leaflet. They both work now for a 20% discount, but you can have 35% if you join UFAW. So once again, please join UFAW. We'd really love to have you as a member, and we'll try and make it worth your while with a discount on our books and so on. And we really hope to see you next year in Edinburgh, either in real life, if you can make it to Edinburgh, or online. We really love to see you, and we're really confident that we can run a real world meeting next year, barring anything really unexpected. So it would be great to see everyone next year. And finally, just to say a huge thank you to a long list of people, our speakers and poster presenters, everyone who voted in the poster competition. The session chairs have done an amazing job. It's a really tough job to chair sessions at these online meetings, so thank you to them. They've done an amazing job fielding all the questions to the presenters. Thank you to everyone who's been tweeting out there. It's really great to see everyone spreading the message online. Thank you to everyone who came along, and a really special thank you to the UFAW staff who make this meeting possible. There's all sorts of people working away in the background to make sure that these meetings run smoothly, and not least Steve Wickens, who, if I can persuade him, should turn his camera on and accept a thank you from me and hopefully from everyone else, because it's really Steve running all the technology in the background who's make sure that this meeting works really well. So Steve, if you want to pop on screen, do so, but I'm going to applaud anyway. Thanks, Steve.
I'm on, thank you. So hi everyone, thanks for asking all the questions and things, much appreciated.
Yeah, and big thanks to you, Steve, and also thanks to everyone at UFAW, and especially Sam and the folk back in the office who run all the admin and make sure that everyone's registrations and donations go through. That's really crucial too, and thanks to Birte and Liz and all the other scientific staff who've been really central to making this meeting a success. So with that, I'll wish you a nice evening, afternoon, whatever it is, wherever you are, and hope to see you again in a year for another really exciting meeting. Thank you very much.